Good day everyone. Welcome to the course on next generation sequencing technologies, data analysis and applications. In the last few classes, we have talked about read mapping, we talked about the algorithms and we have seen how we do the read mapping hands on. So, once we have done the read mapping, the next step is to do variant calling. So, this is what we will be focusing on today. So, the agenda for today's class is that we will discuss the different variant types that are possible, and we will actually pick only certain types for this class, and then in the subsequent classes we will look into other variant types and how we can call these variant types from the map data. And then we will also talk about some of the applications of variant calling, right. So, this will help you understand, right, why this is a very important problem. Okay, so, let us begin and start with the different variant types. Okay. So, these are the keywords that will come across today. So, variants, SNP and indel. Going back to the flow chart for NGS data analysis, we have looked into different types of data in different data formats from different sequencers. Then we have talked about data quality control and we have done that hands on. And in the last few classes, we have talked about read mapping against a reference sequence. So, once you have done this read mapping, there are three applications that you can see, three types of analysis that you can do after read mapping. One is genome analysis. So, this involves identification of single nucleotide polymorphism or SNP, indels, so insertions, deletions, especially small ones, and then we also have structural variants. So, we will talk about each of them one after another starting from today. So, these lines actually mark where we are now. So, we have learned about this mapping, and also on the other side we have learned about the quality control. Up to that point we have reached, but we have not talked about the assembly. So, we will now focus on this genome analysis, which is highlighted here. So, the problem of identification of these variants is called the variant calling, okay, and there are different types of variants that are possible. You have SNP or SNP, we have indels, we have CNVs and we have SVs. Okay. So, what are these SNV, SNPs? So, these are the full forms, full expression. So, single nucleotide variation or SNV, then you have single nucleotide polymorphisms or SNPs. Then you have insertions and deletions, which are called indels in short, and we have then copy number variations or CNVs, and we have structural variations or SVs. Okay. So, we will come across all of these one after another, but today's discussion will be mostly focusing on these SNP, SNPs, and I will also introduce these SVs and CNVs in very brief. Okay. So, what is variant calling? So, variant calling is the process by which identify these variants from the sequence data that we have against a reference genome sequence. So, we need the reference data, we do the mapping and then we do the variant calling. Okay. And as you can probably understand from this, this is a comparative analysis, which means we do a comparison of a target genome against a reference genome. Okay. So, let us now discuss these different variant types. What are the major variant types that we can see when we do this variant analysis for different types of genomes, right. So, researchers have done this kind of analysis for a long time now, and they have seen different types of variants, which are actually quite common when you do this kind of analysis. So, we also call them as mutations, as you are familiar with this term. So, the first variant type is the SNV or SNP. Okay. So, here is a very small example of the reference genome, as you can see on top. So, we have the reference genome here, and then we have the target genome or the genome that we are working with, and we have mapped against the reference genome. So, here you can see this space in blue that is actually different from the reference genome, right. So, and this is what we call as SNV or SNP, right. So, single nucleotide variation or single nucleotide polymorphism. Now, you can have not just one such change, you can have multiple changes in nearby regions, and we call them as MNVs, right, multiple nucleotide variations. These are very kind of small number of changes, right, maybe 3, 4 at most, and not more than that. So, we have used these two terms, right, SNV and SNP, and you might be wondering why, what do these two terms mean, are they same? So, we use these terms actually often interchangeably, and people think like they are same, but there is a slight difference. But again, it depends on the person you are asking. Okay, so there are again different conventions depending on the researcher. So, some will call SNP as a variant that is present in at least 1% of the population. So, let us say you are studying a variant like this, and you observe this variant in some individuals in the population, right, whether this variant is present or not, and then you calculate the frequency. Okay. And if that frequency is greater than 1%, then you call this as a single nucleotide polymorphism, right. So, this comes from a population genetic perspective. But on the other hand, there is another convention. So, SNP is used for germline variants and SNV is used for somatic variants. Okay. So, what are these germline variants, somatic variants? We will discuss in a moment in the next slide. But again, so how do you know which meaning is being used? So, this is something that again you would have to ask the person who is using these terms. So, again, when you are using these terms, it is actually better to define them yourselves. What do you mean by SNP and or what do you mean by an SNV? Okay. So, because of this confusion that you have out there, it is better to define that before you start writing or analyzing anything with SNP or SNV. So, now the question is this SNV or SNP? Again, as I said, this depends on the convention, depends on the researcher that you asked about. Okay, so, we have introduced these two terms, germline and somatic variants. So, what are these germline and somatic variants? So, let us define those. So, germline variants are variants that are inherited from parents. So, as you know, right, in the zygote we have one copy of chromosome from the father, one copy of chromosome from the mother, right, and these variants actually are inherited from the parents. So, these will be present in the zygote and they will be subsequently present in all cells that come out that are arising from the zygote. Somatic variants on the other hand, these are variants that are generated during the development process, during divisions of the cells from the zygote, right. So, it is not, these variants are not present in the zygote, they are originating during the cell division process, right, because these changes are happening, these mutations are happening. So, these variants do not affect germline and that is why they are not inherited. Okay. So, if you see, let us say you sequence genome from tissue samples, some tissue might have these some variants, whereas other tissues might not have those variants, right. So, it is because again these tissues originated from different cells and that is why some cells have these variants, some cells do not have those variants. Okay. So, I hope this is clear what are germline variants, what are somatic variants. So, in our body germline variants will be present in all cells, whereas somatic variants will be present in only some certain cells or certain tissues, right. So, and again these variations can be different from one tissue to another. Okay, so, how do you call these somatic and germline variants, right. So, this is important when you are doing the variant calling, how do you differentiate between these two. So, we can actually remember the definition that we just used. So, let us look at this, like how do you actually call somatic variants. So, in somatic variants what you will see is that these, so if you consider these 4 cells, if these 4 cells have different variants in these certain positions, right, these variants will be called as somatic variants. Okay. So, you can see this, we have kind of giving examples of 2 positions here, and in one position cell 1, cell 2 they have this A, right, compared to the reference sequence, this is a change, right, that is what we are assuming, and cell 3, cell 4 they show C base. Okay. So, again this is different between this cell 1, cell 2 and cell 3, cell 4, and similarly if you look at the other position, again you have difference, right. So, you have C, T or G. Okay. And this arises because these cells perhaps come from different clones, right. So, what do I mean by this different clonal origin? Okay. So, during the cell division process, so you can imagine this. So, you have this cell, one cell dividing into 2, right, and this will again go on, right. Now, imagine this mutation process or this variant arises here in this cell. Okay. This cell gets this mutation. Okay. So, what we will see is that this variant will be present in this one as well as this one, right. So, they are originating from this cell. So, these two cells here, they will also carry the same mutation, whereas this other two here, right, at the end here, they will not carry this mutation, right. So, they are arising from two different clones. So, that is why you see this difference, okay. And this is happening inside our body, right. These cells are originating, there is this cell division happening, right. So, some cells will have certain mutations, whereas the other cells will not have this mutation, okay. So, that is why you will see these different variants for the same position within an individual, okay, and this is called somatic variant. Okay, so, somatic variants are very often seen in cancers and tumors because there is a lot of variant mutation process going on. So, they generate a lot of variants, and again these variants are originating from different clones inside the tumors. So, you will see this lot of these somatic variations in tumor cells if you sequence them. So, now moving to germline variants. So, you can probably now understand how we will identify these germline variants. So, germline variants, they will show the same mutation compared to the reference sequence. So, if you take all cells, they will show the same mutation. So, again we are taking these examples of two positions, and we are assuming these are different from the reference sequence, and you can see all cells show A in this position and G variant in the other position, right. So, this is because of the reason that they are originating from the same clone, right. So, as I mentioned, these cells originate from these variants are present in the zygote. So, they are originating from the parents. So, this cell will have this variant present, right, and it is dividing then, okay. And what this means, all cells that are coming from this one here, right, they will be carrying this variant or this mutation, okay. So, you can now imagine, so the zygote, so at the single cell stage, this mutation, this variant is present, and as the development process is going on, the cells are generated, right. So, you are getting this cell division. So, all the cells originating from this one will have these mutations, okay. So, that is why these are called germline variants. So, these will again be passed on to the next generation. Okay, so, now we come to the next variant type, which are small indels. So, we can see insertions and deletions. So, let us talk about insertions. So, we are talking about small insertions and deletions mostly, right. So, you can have very large scale ones that will actually come under the structural variations. So, this small means 8, 10, maybe 8, 10 base pairs or at most, not more than that. So, what is insertion? So, again we can take this example of reference genome and target genome. So, target genome means the genome that we are sequencing or that we are analyzing. So, in red you have the reference genome, and in blue you have the target genome, okay. So, insertion means there is a sequence, DNA sequence that is present in the target genome, right, and not present in the reference genome. So, you can see this blue region here, this is extra in this target genome, right, and it is not present in the reference genome. So, this extra sequence has been inserted into the reference genome, okay. So, that is why it is called insertion. Then you also have something called deletion, and this looks like something like this. So, we have the reference genome in red and the target genome or the sample genome in blue. What happens in deletion is that this region from the reference genome is not present in the target genome, right. So, this region is deleted in the target genome, okay. So, this is what we call as deletion. Then you also have the structural variants, which we will actually talk about later, and under these structural variants we have copy number variations, we have non-CNV structural variants, and we also have complex SVs. Okay, so, we will talk about these different types of structural variants later on when we go to the next classes. So, just very briefly, what are copy number variations? So, we have something called duplication. Now, this duplication can be gene duplication. So, where a gene is duplicated means it is supposed to be present in one copy, but instead of that it is present in two copies. Okay. So, this is what we call gene duplication, or you can have segmental duplication. So, what segmental duplication means? It involves multiple genes or a larger region of the genome that is duplicated, okay, instead of a single gene. And similarly, you can have deletion. So, again do not confuse this with the indel ones, these are large scale deletions. So, which means you have a gene or a segment can be deleted. So, just to illustrate this copy number variation, right. So, you can imagine this is a region, a gene or a segment of the genome, right. In the red, this is the reference genome, and in the blue target genome, you have present this region present in 2 copies. Okay. So, this is what we will call as duplication. Okay. So, the general term for duplication is amplification. So, where you can see even more than 2 copies of the same region. Okay. So, this can happen that you have 3, 4 copies of the same gene in the target gene. Okay. So, that will call as amplification. Whatever deletion you can probably guess, right. So, let us imagine you had these 2 regions, right. So, 2 copies of the same gene that was present in the reference genome, but 1 copy is missing in the target genome, right. So, this actually is deleted, 1 copy is deleted. So, you are left with just 1 copy in the target genome. Okay. So, this is what we will call as deletion. Again, this can happen for a single gene, or it could be multiple segments that are deleted. Then you have different SVs, right. So, we will talk about them later on, not in this class. Inversion, translocation and complex SVs, right. So, complex SVs are actually combination of these copy number variations and other types of SVs. Okay. So, this we will discuss in the next classes. Now, we will introduce a couple of terms that we often use when you are analyzing these variants. So, one is called targeted resequencing. So, what we do in targeted resequencing is instead of sequencing the full genome, we only target a subset of genes or the regions of the genome that we sequence. Okay. So, why would you do that? So, this is important. Sometimes what you will see is that these regions are actually of interest to us. Now, these targeted regions, they could be exomes, right, mostly the protein coding portion of the genome, or it could be some specific gene of interest, right. So, let us say you are genotyping patients and you know that couple of genes are involved in certain diseases, right. So, they can give rise to certain disorders. So, in that case, if you are screening patients for that disease or disorder, you will sequence only those couple of genes, right. And this is actually very effective, because this is faster, right. Instead of doing the full genome, you are just sequencing only a small part of the genome, and also cost effective, right. If you need only sequence of only a part of the genome, you just sequence that region. And also one advantage of targeted resequencing is that you can actually do deep sequencing of that region to identify rare variants. Okay, so, what are these rare variants? So, imagine you have in population, let us say in 100 individuals, only let us say a couple of individuals or 2 individuals carry this variant, right. So, this will be a rare variant, right. Only a small, very small number of the population is carrying this variant. So, once you do deep sequencing, you can identify this rare variant in the populations. Similarly, there is a related term which is called amplicon sequencing. Okay. So, this is again related to targeted resequencing, where we actually amplify only a specific region of interest and then we sequence that region. Okay. And again, this is a focused analysis, we are looking at only 1 to 2 genes maybe, or a small segment of the genome, and we do deep sequencing. So, this again helps in getting to the rare variants. So, we will now talk about some of the applications of variant calling before we actually jump into the variant calling pipeline in the subsequent classes. This will give you a better understanding how variant calling is important. So, one of the first applications is clinical cancer genome profiling. So, what do you mean by this cancer genome profiling? So, as we have seen, as I have discussed, right, in tumors you have a lot of these mutations accumulated, right. So, this is something that we see, there is a lot of genetic variants present, right. So, once we want to, what we want to do in cancer genome profiling is that we want to study these variants, right. What are these variants that are accumulated inside tumor cells, because some of these variants might be important for some of the phenotypes of cancer, or maybe it might help in immune evasion, or it might help in drug resistance or therapy resistance, right. So, you have right, many times the therapy, anti-cancer therapy does not work on a tumor, and we now know, right, there are some variants that kind of are responsible for this resistance. Okay, so we can study this germline and somatic mutations from tumor, right. So, as I have told that tumors actually accumulate a lot of somatic mutations during the cell division process, and we can study these somatic mutations, and by studying them we can actually identify the driver mutations, right. So, what are the driver mutations that are actually driving the progression of tumor through different stages and driving those phenotypes, right, the drug resistance or immune evasion phenotypes, etc. We can identify different subtypes of tumor. Different subtypes will show certain different characteristics, and they also respond to different therapy. So, by studying these variants we can actually do something called subtype classification, and that can help in selecting the appropriate therapy, right. So, there is this term we have introduced earlier, which is called precision medicine. So, in the first few classes, right, we talked about applications of NGS and we talked about precision medicine. Precision medicine means we are developing therapy depending on the genotype, right. So, if certain mutations are present in tumor, we give certain therapy. If the other types of mutations are present, we give other type of therapy, right. So, this kind of variant calling process is very important if we are doing this type of identifying the genetic basis of these diseases and also identifying the therapy. So, here is a paper that you can read, and which discusses about this validation of this clinical cancer genome profiling based on DNA sequencing methods, and this will give you a good overview of the whole process. We can also study cancer mutational signatures. So, what do we mean by this? So, here is an example of mutational signatures. So, we see certain different types of mutations in cancer, and we study them in the context of the genomic sequence. So, what you see here, we have these different types of mutations C2A, C2G, etc., and what we are looking at is the frequency or the number of occurrences of these mutations in certain types of cancer. Okay. So, this is for one type of cancer, right, and as you can see, we see a lot of this C2D mutation, and we also have a lot of T2C mutation, right. What else you can observe here is that this C2T mutation or T2C mutation, they occur in certain context much more often than other, right. So, for example, you see here this T2C mutation is the occurrence is much higher if you are looking at this AA context, right. So, you have this ATA sequence, and this T is often mutated to C. Okay. So, again we can study these context specific occurrences, and we can identify the different mutational signatures that are present in different cancers, and again we have a lot of data now, a lot of analysis has been done already, and we know these mutational signatures for different types of cancers across different patients. We can also identify germline variants that are present in individuals, right, by just comparing parents genome to the child genome, right. So, if you are studying one individual and you compare the variants that are present in the parents, you can identify what are the germline variants, and of course, then you can also identify what are the somatic variants. Now, one important application of identifying germline variants is actually to identify variants that are associated with genetic diseases. So, there are several genetic diseases for which we know like different types of mutations in certain genes are responsible. So, by analyzing these variants, right, we can actually associate some of these variants to genetic disease, right. So, we can imagine, right, you have a situation where you have specific variants we see associated with a disease, right. So, what I can do, I can draw something like this. Okay. So, let us say you have this is the frequency, right, and this is the disease phenotype, right, and maybe this is something, the distribution looks something like this. Okay. And some of these mutations, right. So, frequency of individuals, right, they are showing this different disease phenotypes, and we see certain mutations are associated with very severe disease phenotypes. Okay. And that might mean, okay, these mutations, these variants are perhaps associated with this genetic disease. Okay. So, studying these germline variants can help us also study these genetic diseases. Okay. So, here is again some reference for your better understanding. You can go and read looking at the disease gene discovery, right, association of certain genes, mutations in certain genes with certain genetic diseases. Okay. So, another application of this variant calling is that we can study genetic diversity in populations, and we can infer population history, how the population has evolved, right, what kind of changes have happened, and we can also associate it them with certain events in the history. Okay. So, for example, we can study the population migration through different continents, etc., when you can study this genetic diversity. We will not go into it, but you can of course explore more about this. We can also study different cell type diversity using next generation sequencing variant application, variant calling application, right. So, we can for example study T cell receptor diversity. Again, this is very important for identification of different pathogens. So, T cell as you probably know from immunology is that the receptor varies a lot, and this helps in identification of certain pathogens. So, with this next generation sequencing and combining with variant calling, you can study this T cell receptor diversity in great detail. Okay. So, here is again a reference for you to read, which actually talks about these T cell receptor sequencing technologies and trying to quantify this diversity. So, here are the references that we have used for this class, and to summarize, we have talked about different types of variants, but we have focused mostly on SNV, SNP and small indels in this class, and there are many more out there, the structural variants especially, which we will talk about in the next few classes. Okay. So, we will come to the structural variant analysis, and then we will discuss that in much more detail. So, very briefly we talked about copy number variations or CNVs, but apart from that we also have non CNV SVs, structural variants, and we also have complex structural variants, which are combination of CNVs as well as non CNV SVs. Okay. So, we will talk about that in much more detail in the next classes when we talk about SVs, but this is something probably that is clear for you, right, that there are a huge number of variants that we need to identify when we are analyzing genome sequencing data. Then we talked about these variants and the applications of these variants across different fields, right. So, we have shown you that there are different applications, and we have very briefly discussed some of these applications, but I encourage you to go into more detail, read those references, and you will get a better idea of these applications. And we also talked about the clinical implications of this, right. So, we can apply variant calling for clinical applications such as the mutational signatures in cancer or disease gene discovery for genetic diseases. Thank you.